Hi everybody, let us discuss some of the questions from transcription topic of the chapter Molecular Basis of Inheritance. Let us enter into the questions. First one. Choose the correct statement. Always only one of the strands of DNA has information for the formation of all the proteins of an organism. No, this is wrong. Some information related to the formation of the proteins is present on one strand of the DNA, and information for the formation of some other proteins present on another strand of the DNA. But at a given place, information for the formation of two different types of proteins is not present in the opposing strands. Right? So, first statement is wrong. All transcriptions lead to translation. No. Only the transcription of messenger RNA leads to translation. Transcription of transfer RNA and ribosomal RNA, snRNAs, etc., microRNAs and all that. So, those transcriptions do not lead to translation. Complete stretch of DNA strand is transcribed at a time by the activity of RNA polymerase. So, complete stretch of the DNA strand is not transcribed. Only a part of it, where a particular gene is present, that one only transcribed. Right? In a DNA molecule, several genes are present, but at a given time, only one gene is transcribed. So, third option is also wrong. Second option is also incorrect. If one strand at a place of DNA is transcribed at one time, other strand of DNA may be transcribed at other place of the DNA. This is absolutely true. The fourth option must be the correct one. Let us see the answer. Fourth one. For some genes, one strand has information for a transcription, and for some other genes, the other strand of DNA has information for a transcription. All transcriptions do not lead to translation. Transcription of messenger RNA leads to translation, but transfer RNA and ribosomal RNA transcription doesn't lead to translation. Only a small stretch of DNA and only one of the strands is transcribed at a given place by the activity of RNA polymerase. In different places, different strands of the DNA are transcribed for formation of RNA. Coming to the next question. This is matching. In this matching, need for initiation of transcription. What is required? Sigma factor is required. A2. Site where RNA polymerase is bound. RNA polymerase is bound initiate the promoter region. B3. A2, B3. Actually transcribed region, that is structural gene. So that region where information for the formation of the protein is present, or information for the formation of transfer RNA or ribosomal RNA is present. So C4, usually needed for transcription termination. The O factor is required, 1, D1. So A2, B3, C4, D1. So second option must be the correct one. Let us see the answer. Second one. So needed for initiation of transcription in prokaryote, that is sigma factor. Site where RNA polymerase is bound, promoter of transcription unit. Actually transcribed region, structural gene of transcription unit. Usually needed for transcription termination, that is Rho factor. Coming to the next question. Choose the correct statement with respect to transcription unit. Transcribed DNA strand is named as coding strand. It is a template strand or anti-coding strand. So coding strand is not transcribed. The strand of DNA with 5' 3' polarity is called template strand. No. The strand of DNA with 3', 5' polarity is called a template strand. So second is also incorrect. The sequence of nucleotides in template strand is similar to the sequence of a formed messenger RNA. Never. The sequence of nucleotides in template strand is complementary to the sequence of formed messenger RNA. Third one is also incorrect. The promoter of transcriptional unit is present at 5' end of coding strand. Absolutely true. So the sequence of the promoter, position of the promoter, terminate, etc. is decided with respect to the 5', 3' polarity strand. Right? So fourth option is the correct one. Let us see the answer. Fourth one. Transcribed DNA strand is called template strand. The strand of DNA with 5', 3' polarity is called coding strand. The sequence of nucleotides in template strand is complementary to the formed messenger RNA. The promoter of transcriptional unit is present at 5' end and terminated at 3' end of coding strand. Coming to the next question. The statements are given. You have to choose whether statement 1, statement 2 are correct or incorrect. If correct, which one is the correct, which one is incorrect. That you have to choose. Additional regulatory sequences of transcriptional unit can be present downstream to promoter. Absolutely. Additional regulatory sequence can be present downstream or upstream to the structural gene or promoter. So, operator is regulatory sequence of an operon. So, this is also true. In case of prokaryote, the operator is the regulatory sequence. Operator is not transcribed, but helps in regulation of transcription. So, statement 1 is correct. Statement 2 is also correct. Both statements 1 and 2 are correct. Fourth option must be the correct one. Fourth one. Additional regulatory sequences are present either upstream or downstream to the promoter. Operator is additional regulatory sequence or transcriptional unit of lac operon present downstream to promoter. Downstream is toward the 3' end. Coming to the next question. Choose the incorrect statement. Gene that has information for the formation of ribosomal RNA is a structural gene. Yes. So any gene that has got information for the formation of messenger RNA or ribosomal RNA or transfer RNA, other RNAs is a structural gene only. Right? This is the correct one, but we want the incorrect statement. Genes that are transcribed into transfer RNA are structural genes. Absolutely true. A cistron is a structural gene. Absolutely true. Operator is a structural gene. Wrong. Operator is not a structural gene. Operator is a regulatory sequence. In loose sense, they can be called as genes, but it is regulatory sequence operator. Operator is not transcribed. It helps in regulation of transcription. The fourth option is the incorrect statement. It is not a structural gene. The structural gene is that gene which is transcribed. The fourth option is the incorrect statement. Fourth one. Operator is not a structural gene as it is not transcribed but regulates transcription. It is a regulatory sequence. Genes which have information for the formation of protein, ribosomal RNA, transfer RNA and other RNAs are called structural genes. Promoter, operator and terminator are not structural genes as they are not transcribed but involved in regulation of transcription. Coming to the next question. The assertion reason type. Assertion. Lac I. That's the regulatory gene of lac Lac I is monocystonic structural gene. Absolutely true. So lac I, upon expression of this one, lac I forms the repressor protein. So a cystron is that portion of the gene which has got information for the formation of proteins. Monocystron has got information for the formation of only one type of protein. The lac I is monocystonic structural gene only. It has got information for the formation of only one type of protein, that is repressor of lac operon. Monocystonic structural genes are usually present in eukaryotes. This is also absolutely true. But assertion it is not explaining. So A and R are correct. R is not correct explanation to A. So fourth option must be the correct one. Let us see the answer. Fourth one. Lac I is monocystonic structural gene associated with Lactoperon of E. coli, as it has information for the formation of only one type of protein, that is lac repressor, but it is not a split gene. Monocystonic structural genes are usually present in eukaryotes. They are seldom present in prokaryotes. Coming to the next question. Genes that have exons and introns are named as. So interrupted genes are also known as split genes. So if I got exons means expressed sequences, introns intervening sequences. Expressed exons or expressed sequences have got information for the formation of protein. Introns do not have any information for the formation of proteins, or their information doesn't indicate any amino acids. During processing of messenger and intron regions are removed. Both exons and introns of the split gene are transcribed, but only exons are translated, introns are removed during processing of messenger RNA or heterogeneous nuclear RNA. Split genes. Option 1 is correct. Regulatory sequences, no, they are transcribed. Monocystrons, right? So a monocystron now the information for the formation of only one type of protein. The monocystron may be split gene, it may be non-split gene. If exons and introns are there, it must be referred as split gene. Polycystrons, no. Polycystrons have information for the formation of more than one different types of proteins, usually related to a particular biochemical pathway or metabolic pathway. So option 1 is correct. Let us see the answer. 1. Genes that have expressed sequences known as exons and intervening sequences introns are called split or interrupted genes. Regulatory sequences are associated with structural genes which are not transcribed. Monocystrons are genes that have information for the formation of only one or single type of polypeptide or protein. These are common in eukaryotes and uncommon in prokaryotes. Polycystrons are those genes that have information for the formation of more than one type of proteins and transcribed into single messenger RNA. These are common in prokaryotes. Coming to the next question. Identify the correct statements. There is only one type of RNA polymerase in prokaryotes. Absolutely true. That is a correct statement only. There are 3 or more types of RNA polymerases in eukaryotes. Absolutely true. The RNA polymerase 1, RNA polymerase 2, RNA polymerase 3, and even the chloroplast have got mitochondria with different RNA polymerases. So there are 4 or 5 types of RNA polymerases are present. But in the book it is given as 3. But the additional information is 4 and 5 may also be present in eukaryotes. So B is also correct. Ribosomal RNA of eukaryotes is formed by the catalytic activity of RNA polymerase 1. Absolutely true. As well as RNA polymerase 3. So RNA polymerase 1 forms 28S ribosomal RNA, 5.8S ribosomal RNA, and 18S ribosomal RNA. RNA polymerase 3 is involved in the formation of 5S ribosomal RNA, besides transfer RNA and smaller nuclear RNAs. C is also correct. RNA polymerase 2 is involved in transcription of mRNA of eukaryotes. So RNA polymerase 2 is involved in transcription of messenger RNA of eukaryotes. Absolutely true. RNA polymerase 3 is involved in transcription of transfer RNA and snRNAs of eukaryotes. Absolutely true. So A, B, C, D, E, all the 5 are correct statements. A, B, C, D, and E. Let us see the answer. C. Prokaryotes have only one type of RNA polymerase. 3 or more types of RNA polymerase are present in eukaryotes. 28S, 18S, and 5.8S ribosomal RNA are formed by RNA polymerase 1, and 5S ribosomal RNA, snRNAs, and transfer RNAs by RNA polymerase 3 of eukaryotes. RNA polymerase 2 is involved in the formation of messenger RNA, siRNAs, the smaller interfere RNAs of eukaryotes. Coming to the next question. Arrange the following events in correct sequence during transcription and processing of messenger RNA of eukaryotes. The fresh transcript that is formed by the activity of RNA polymerase 2 in eukaryotes is known as heterogeneous nuclear RNA. So during transcription it is subjected to some processing, and after transcription also it is subjected to some processing. Let us see what is given. Capping of mRNA. Capping of mRNA takes place at the time of elongation of transcription. Initiation of transcription, this is the first one. Termination of transcription. So at the terminated region, at the end, termination takes place. Splicing. Splicing takes place only after formation of heterogeneous nuclear RNA. Tailing of messenger RNA. It takes place soon after the transcription of heterogeneous nuclear RNA. The first one is B. B should be the first. Initiation of transcription. When the transcription is in progress, there is a capping of messenger RNA. During capping, 7-methyl guanosine triphosphate is attached to the 5' end of transcribing messenger RNA or heterogeneous nuclear RNA. BA. Next one is termination of transcription. Transcription has to be terminated. So BAC. Next, tailing of messenger RNA. During tailing of messenger RNA takes place at the 3' end of the RNA that is formed, or messenger RNA, heterogeneous nuclear RNA that is formed. During tailing, there is additional several adenine nucleotides, about 250 to 300, or sometimes 50 to 300, right? Adenine nucleotides are attached during tailing. After tailing, then only splicing. During splicing, the introns of the transcript are removed and only exons are attached, and it is converted to active messenger RNA. Their capping gives must for effective binding of messenger RNA to ribosome. Right? Tailing is must for proper transport of the messenger RNA to cytoplasm from nucleus. Splicing removes the non-coding region of the cistron. Right? That is, introns are removed, only exons are attached. So that region which has got information for amino acids is attached, which is not having information for amino acid sequence is removed. So splicing takes place at the end. So option 2 is the correct one. Let us see the answer. 2. The various stages during formation and processing of eukaryotic messenger RNA are initiation of transcription, capping of messenger RNA during elongation, termination of transcription, tailing of messenger RNA, and last splicing. Coming to the next question. The unusual nucleotide 7-methyl guanosine triphosphate is added to the messenger RNA of eukaryotes during capping is added. The addition of 7-methyl guanosine triphosphate at the 5' end of the freshly transcribing messenger RNA is known as capping. So besides this adding of 7-methyl guanosine triphosphate, the second and the third nucleotide ribose sugar is also methylated. Sugars are also methylated. That is also done. So that the mRNA is well protected from exonucleases. So that is for capping of messenger RNA. Let us see the answer. Not tailing, not transcription termination, not transcription initiation. Capping of messenger RNA. Fourth one. 7-methyl guanosine triphosphate is added during capping at 5' end of transcribing messenger RNA at the time of elongation stage of transcription. Sugars of second and third nucleotides of messenger RNA are also methylated at the time of capping. During this, there is a formation of 5' phosphodiester bond between 7-methyl guanosine triphosphate and first nucleotide that has come during transcription initiation. That is, please like, comment, share, and subscribe. Thank you.